Good morning, everyone. I'm Meenakshi Jain, Head of Development Regulatory Center, Sandoz Development Center from India. It is my pleasure to be part of the session 6, Implementing GDUFA Science in Product Development and ANDAs, and share some insights from an industry perspective.
Let me share with you the disclaimer first. This presentation is based on publicly available information, and the views presented are the views of the presenter and not those of Novartis or Sandoz. The agenda for today's presentation is Reflections for GDUFA Science and Research, Challenges and Opportunities for some of the complex projects, and Way Forward for implementing GDUFA Science early in development. In this slide, we will talk about reflections of GDUFA science and research. Certain pharmaceutical products are harder to develop as generics, and these complex products have few generics or none at all. In the absence of market competition among generic alternatives, these medicines can be so expensive to patients who may not be able to afford them. The outcomes from GDUFA-funded research expand understanding of these complex products and often contribute to the development of advanced methods to characterize product quality and performance. The GDUFA research outcomes also prepared FDA to assess ANDAs referencing complex products which ultimately improve patient access to complex generics that were presumed to be unfeasible to develop even just a few years ago. In front of you are some of the reflections of GDUFA science and research already conducted in 2021 for complex injectables, formulations and nanomaterials, complex mixtures and peptide products, immunogenicity risk assessment, and ophthalmic products. Based on these research outcomes, first FDA approval for generic glucagon for injection and difluprednate ophthalmic emulsions were received. As part of FDA's commitment to expanding its collaboration and communication with industry, Center for Research on Complex Generics was established to enhance how generic industry stakeholders and FDA work together to overcome challenges impacting patient access to high quality, safe and effective generic products.
Coming to the next topic, oligonucleotides, API sameness, and immunogenicity assessment.
Oligonucleotides are short synthetically derived RNA and/or DNA strands that influence gene regulations. Synthetic oligonucleotides are considered small molecules regulated as drugs by CDR and follow a generic regulatory pathway. Various classes of oligonucleotides exist like antisense oligonucleotides, small interfering RNA, aptamers, etc. There are additional challenges for generic oligonucleotide drug development. There are no FDA guidelines that specifically address the quality aspects and expectations. Most oligonucleotides are solutions for injections, and bioequivalence may be considered self-evident. Hence, biowaiver is possible provided API sameness is established and impurity profile of test and R&D is comparable. 3 batches of the test product and the R&D is required for the API comparison and the sameness study, and impurity characterization by orthogonal methods is required as most impurities exist as mixtures of closely related molecules and many impurities co-elude with the active ingredient. It is also required to include information to justify that any impurity difference in the generic do not give rise to unknown differences in immunogenicity or toxicity. However, there is lack of clarity and we are required to contact FDA for further questions. To address these challenges, FDA is actively working on characterization of synthetic oligonucleotides to support generic drug equivalence. FDA has developed HRMS MSMS methods for sequence characterization and impurity analysis of synthetic oligonucleotides and product related impurities. and also issued a product-specific guidance for nusinersen sodium injection in February 2022. FDA is also planning to publish 6 additional product-specific guidances in the near future.
For a comprehensive characterization and demonstration of sameness, FDA recommends that potential applicants develop and use appropriately validated orthogonal methods to perform side-by-side comparison. Primary sequence of oligonucleotide in the test API can be controlled through each elongation cycle in the API synthesis. Due to stereochemistry at the phosphorus chiral center of phosphorothioate linkage, nusinersen contains many different diastereomers. The expectation is to measure R by S configuration ratio at each phosphorothioate nucleotide linkage following each elongation cycle using appropriate methods. So, approach for demonstrating the sensitivity, discriminatory and resolving power of analytical method for diastereomery composition analysis should be appropriately justified. Hence, we request FDA to do further research on analytical methods for demonstrating comparable diastereomery composition of test product and RLD, and also sequencing and impurity identification and quantification by HRMS requires advanced software, so the current available software is not validated and can be used for research purpose only. So we would like FDA to collaborate with software providers and recommend some specific softwares. Further, there are no general guidances available for reporting identification thresholds for oligonucleotide related impurities. Reporting threshold directly impacts analytical method development. Therefore, it would be highly appreciated by industry to provide directions for reporting thresholds. As per nusinersen product-specific guidance, generic applicants are advised to contact FDA for questions related to immunogenicity and inflammatory risk assessment and comparability of impurities. So in general, antisense oligonucleotides are relatively small, contain fewer potential epitopes than their larger protein counterparts and thereby are less likely to generate immunogenicity. DNA is generally thought to be relatively non-immunogenic. A risk-based assessment on antisense oligonucleotides published in the scientific literature showed a low potential for antisense oligonucleotide-induced immunogenicity based on the route of administration, antisense oligonucleotide structure, and treating disease type. Also, an ANDA relies on FDA's finding that the previously approved drug product, that is the reference listed drug, is safe and effective. So we believe that immunogenicity potential of oligonucleotides is low. Hence, we request FDA to do active research and provide guidance on how immunogenicity of generic oligonucleotides should be addressed and what studies should be performed if needed. If FDA can provide guidance on clear immunogenicity risk assessment pathways which we can follow, and if there are any in silico tools available for assessing immunogenicity potential for generic oligonucleotides. Coming to the next topic on complex products. API sameness study is a requirement for complex APIs, for example, metal complexes, peptides, oligonucleotides, and synthetic polymers. There are different expectations for DMF review versus ANJ review as well as for different products. While on one hand, the expectation is to demonstrate API sameness by minimal manipulation of intact RLD. However, in some of the products, the expectation was to extract the API from the RLD and compare with the pure API during the DMF review. The drug product manufacturing process, for example, terminal sterilization, can have impact on the API properties. We believe that extensive characterization of DMF drug substance shows an appropriate quality and consistency in drug substance manufacturing process, while physicochemical property sameness study of test drug product and RLD should be sufficient as patient is receiving the final drug product. So we request FDA to do some more research in this direction, and if they can work on some specific case studies and provide clear directions in product specific guidances or provide general guidances for the sameness study. Also, there are no guidances on the acceptance criteria for demonstration of sameness for complex products. If FDA can explore and provide guidance how to set the acceptance criteria for demonstration of sameness for complex products would be very beneficial. There are no guidances on extent of method validation qualification requirement for characterization methods which are used for the sameness study and comparability testing. Can FDA publish guidance on extent of method validation qualification for different types of characterization methods which are advanced techniques? Some of the techniques are listed here. Coming to the next topic for ophthalmic products. So in financial year 2021, research efforts continued to address challenges in three areas: development of in vitro release test methods that more closely resemble in vivo physiological conditions, identification and characterization of critical physicochemical properties of complex ophthalmic products in vitro, ex vivo, and in animals, advancement of in silico modeling to investigate the impact of formulation properties on ocular PK and PT. So, a new IVRT method called adaptive perfusion was developed employing principles of tangential flow filtration. So, the method was also published in the Journal of Controlled Release. Also, FDA is actively working on these areas in this financial year.
While adaptive perfusion method would be useful for industry, it might not be suitable for all ophthalmic products, and industry might not have access to these techniques. So the development of novel in vitro release test methods that more closely resemble in vivo physiological eye conditions might not be feasible. It is not clear to what extent should in vitro release method exhibit physiological conditions, and should the discriminatory capability of IVR method be relevant to all critical attributes of the drug product. So we would like FDA to do more research on other in vitro methods, for example, France diffusion cells, immersion cells, or transfer method or any other which are relevant enough for in vitro release of ocular formulations, and also to do more research to identify critical attributes to show the discriminatory capability of IVR method.
Coming to the next topic on impurities. Justification of higher level of impurities observed in generic drug products based on levels observed in reference product is challenging in scenarios where reference drug product lots are not available in the market or close to expiry lots of RLDs are not available. Both these scenarios can delay the access of medicines to the patient. Can FDA accept impurity justification based on statistical simulation of test and RLD, for example, ASAP prime data for small molecules at the time of submission, and supplementing the data with near expiry lot of RLD during the review process? Can FDA accept the justification based on the data from Europe or Canada RLD or any other approved generic products? We would like FDA to make a process for FDA approval with use of ex-US RLD data for impurities as well as the characterization studies. To help generic industry stakeholders implement scientific insights from GDUFA research outcomes in a manner consistent with the FDA's regulatory expectations, we request FDA to make a database of FDA research outcome and make it available to industry, provide access to literature and research articles so that we can implement the GDUFA science early in development. Also, leveraging product development meetings and controlled correspondences early in development would be really beneficial. And if FDA could provide specific inputs and directions during these meetings would be really helpful. Adequacy of tests and methods is normally a review issue. This should be aligned during product development meetings to ensure smooth review process of ANDA and DMF. Finally, an opportunity for real-time communications with FDA would be really beneficial.
These are some of the references used for this presentation. Lastly, I would like to thank all my Sandoz colleagues who had supported me in every step. It is a great honor to have you all attentive and tuned in. I thank you for spending your much valuable time to be present here. Looking forward for the panel discussion.